Okay, welcome back. I want to continue talking about the endocrine system material. Before I said that we're going to look at these micrographs, I explained the process behind how we stain tissue and how we see material, and the basis for why we're going to make decisions about the staining quality of this tissue. We want to look at these 4 tissues, and we're going to do that through the cloud storage files that I have. Now, since the first video, I had to change something. Before, what you saw is what it looked like to you when you log in, but I found actually I was having a little trouble on my computer downloading them. But if I logged into Box, I can do that. So when you go onto this, you will not see this menu on the side and some of the other stuff up here. That's not important. But this way I can pull these up onto the screen. So here is the hypophysis. Now, hypophysis is just another name for the pituitary gland. The word hypophysis comes from the Greek. Hypo means below, and what the hypophysis is below is the hypothalamus, which also has hypo in it. And that's because it's below the thalamus. But it would be a very awkward construction in Greek to call something the hypohypothalamus. So instead, physis is referring to the hypothalamus. It's sort of another name for it in Greek. The hypothalamus is sort of the control center for a lot of physiological processes, a lot of autonomic homeostatic controls. So for the autonomic system and the endocrine system. So physis here comes from the same place as the word physiology. So the hypophysis is what's below the pituitary gland. It is what's below the hypothalamus, which is the pituitary gland. Now, when we talk about the pituitary gland, there's 2 parts, the anterior pituitary and the posterior pituitary. I did misspell it. Sorry, let me spell pituitary correctly. But when we talk about the hypophysis, refer to it as the Adeno hypophysis and the Neuro hypophysis. So one of those means anterior pituitary and the other one means posterior pituitary. And let me just talk about the tissue a little bit to make you understand what's what. So, as I described in the previous figure, what we want to think about here is staining quality, and this is a great place to see that. One side of this picture is darker than the other side. That's all I mean by staining quality, dark versus light, which goes back to what's in the cells that the stain reacts with. The darker staining cells are going to have more RNA in them through having ribosomes and mRNA and that sort of thing, to make proteins, whether it's protein hormones or enzymes to make non-protein hormones. And then light staining tissue is light because it doesn't have all of that protein machinery in it, and it just is going to stain lighter. Now, before we go on, I just want to point out there's this big crack down the middle of the tissue that we see here. This is the natural dividing line between the anterior and posterior pituitary. You would not actually see a physical space between those 2 structures because they're right up next to each other. The pituitary gland is just one big chunk of tissue. But there is a physical separation between them and a little connective tissue holding it together. When you dehydrate the tissue, like I described in the previous video, the tissue shrinks. It's stuck to the glass, so it's not going to just all shrivel in from the outside towards the center, but rather each little part of the tissue is going to shrink to itself. And so the 2 halves of the pituitary gland shrivel up and pull away from each other. And we can actually see that physical separation because it pulls apart at the weak point of the tissue, which is where there's a transition from one type of tissue to another. Glandular tissue to connective tissue to nervous tissue, that sort of thing. So it's going to preferentially crack right there. We'll often see that cracks in tissue fall where there's some obvious line between it. But I just want to point that out here. So one side of this is essentially darker and the other side is lighter. One side is the anterior pituitary, one side is the posterior pituitary. One side is the Adeno hypophysis, the other side is the Neuro hypophysis. So at this point in talking about this in face-to-face class, I'd say, okay, so the difference between these two is what the prefix means. So Adeno or Neuro, what do those mean? And there's certainly going to be more people that are familiar with what Neuro means than Adeno means. Neuro obviously is referring to the nervous tissue, or the nervous system, or the brain, or whatever you want to say. It's related to that stuff. You may or may not know what Adeno means offhand, but if you look it up on Google real quick, it's a combining form relating to gland or glands. So the Adeno hypophysis is a glandular hypophysis, and Neuro is the nervous tissue hypophysis. So as we go back to look at this, one side of this is specialized for secreting things, it's a gland, and the other side is specialized for communication, it's nervous tissue. Now, nervous tissue isn't the only one that does communication, but the way that it does communication is very tissue specific, and so it gives the tissue a particular appearance. So which of these two tissues do you think is glandular tissue, and which do you think is nervous tissue? Which one's going to be making more proteins, and which one's not? So hopefully you've come to the realization that the Adeno hypophysis is the glandular tissue which is making protein hormones. And that's the anterior pituitary. The anterior pituitary is making a bunch of... Oh, that didn't work right. There we go. The anterior pituitary is making a bunch of hormone-based, protein-based hormones, and so it's glandular tissue. It has a lot of RNA and cytoplasm, so it's darker staining, compared to the neurohypophysis, which is nervous tissue. Now, in nervous tissue, you would think you'd see the occasional neuron or something like that. But actually what we're looking at here is fairly neuron-free nervous tissue. This is mostly fibers coming from the hypothalamus, and the cells, the cell bodies, are in the hypothalamus. They make hormones there, and then they secrete them through their axons into the bloodstream here. Those are called neurosecretory cells. They look just like neurons, but they don't synapse on another cell, but instead they release their contents into the bloodstream directly. And so this tissue looks lighter than this tissue because it's basically just a bunch of axons, and maybe some other components of nervous tissue. So they don't stain as darkly. Some of the darker staining stuff in here might be other neurons, they might be blood vessels, I'm not quite sure. This is fairly low power, not enough detail to make out. And it's not what we're concentrating on here. The glandular tissue of the Adeno hypophysis is what we want to concentrate on a little bit more. I think if I go this way, yeah, so this is the same slide. There's a crack between the tissue, and we just see a little bit more detail. We've moved to a higher power magnification, and now we can actually see nuclei. So before we were using the 4x objective, now we're using the 10x objective, so that's the difference between 40x magnification and 100x magnification. And now we can see the detail and make out nuclei. So there are nuclei around here. Some of those might be glial cells. There might be little neurons in here, something like that, but there's not big chunks of gray matter kind of cells. But over here, there's a lot of dark staining, and some of it isn't even nuclei. So you can see how small nuclei are here. So over here, you can see some small dark spots, but then sometimes the dark purple color is just all smushed together and it looks like a big blob. Or the cytoplasm stains a lighter shade of purple, I suppose you'd call it. This is glandular tissue. Now there are some regions that are a little bit lighter in stain, and they are probably not making the actual hormones. The darker staining cells are. Now I do want to point out here, in the end it's not important for this course, but there is a little darker staining strip right here on the edge of the neurohypophysis. And that's because there's actually three regions of the pituitary gland. There's the anterior pituitary and the posterior pituitary, and then this little area in here, I think it's called the medial pituitary, but I might be wrong. And I'm not sure offhand because I usually don't talk about it. It doesn't secrete any hormones that we really care about, and I don't think the textbook we use now, but a textbook I've used in the past, when it referred to that area, it just mentioned the only hormone that's related to it is released only, or was described only in a disease state. So what's secreted from there apparently wasn't even noticeable in normal levels unless you had some sort of disease state. So we don't really pay attention to that part, and so we're just lumping it in here with the lighter staining posterior pituitary. But technically it's its own little thing. And I just want to be completely upfront about that so you don't think I'm waving my hands too much. What we're mostly concerned with is the darker adeno hypophysis and the lighter neuro hypophysis over here. And that difference in staining quality tells us something about this. This tissue is making its own hormones releasing in the bloodstream. This tissue is delivering hormones made somewhere else, namely the hypothalamus, into and delivering them directly to the bloodstream. The posterior pituitary hormones, as they're often called, which again are actually made in the hypothalamus, are oxytocin and vasopressin or antidiuretic hormone. Those are the only things that are released through the posterior pituitary that we cover in this class. And they have effects elsewhere in the body. The hypothalamus makes a number of other hormones. They're released through neurosecretory cells that get into the bloodstream in the infundibulum area, the stalk connecting the pituitary gland to the ventral side of the brain. And then they move through the bloodstream to affect these cells right here. Those hormones that come from the hypothalamus are usually referred to as something releasing hormone or something inhibiting hormone. Like growth hormone releasing hormone or growth hormone inhibiting hormone, or GHRH and GHIH, which basically tells cells in the anterior pituitary to either release growth hormone or stop releasing growth hormone. The particulars of what's going on in that story are covered in the lecture material, but here we can see that. Now, I think I got to get back to so here's the high power look at this tissue. So this is the same tissue we're just looking at, the anterior pituitary. Now we can make out the nuclei, which are darker, and then the nice dark cytoplasm of these cells, and in a number of them we can actually make out the cell membrane. And then there's some lighter areas in between. So these dark staining cells are making these hormones and secreting it into the lighter staining area, which might be little ductlets that carry that stuff to, not ductlets, extracellular space that allows that to travel to the bloodstream. And so we can think of the cells as clustered here. They, to me, look kind of like cobblestones in an old-timey road. Each of these clusters of cells is probably a functional unit. All of these cells make one thing, like growth hormone. And then all of these cells make a different thing, like ACTH, which is a stress-related hormone. And all of these cells might make something else. Now, there are not that many hormones from the anterior pituitary, so in this region, we're probably looking at a few clusters that make growth hormone or one type of signal. But they are clustered together. And the cells all get a signal from the hypothalamus saying release your hormone or stop releasing your hormone. And that's how the hypothalamus controls the endocrine system in a number of the endocrine function units that are discussed in the book. Anyways, so that is the pituitary gland, okay, or the hypophysis. The anterior pituitary is the adeno hypophysis, and the posterior pituitary is the neurohypophysis. Now I want to move to looking at the thyroid parathyroid gland, which is here. So, again, considering staining quality. There's 3 different regions of tissue here. There's this lighter staining area here, this dark cluster right here, and then this stuff that doesn't look like the others. The lighter staining stuff is the thyroid gland. The darker staining is the parathyroid gland, which are both located in the neck. So right at the base of your neck, basically. The thyroid gland is a fairly sizable, spongy-looking piece of tissue that wraps around the anterior and lateral aspects of your trachea. It does not go all the way around to the back. It doesn't meet in the back. So this would be the posterior side of the thyroid. And so on this posterior edge, right where it doesn't meet in the middle, are little chunks of tissue called the parathyroid glands. How many are present in any given person varies. There might be 4, there might be 5, 6, 7, 8. Doesn't matter. The overall amount of tissue is going to be fairly standard. So somebody with 4 parathyroid glands probably has big parathyroid glands. Somebody with 8 parathyroid glands has small parathyroid glands. They don't necessarily have to be attached to the thyroid gland. They might attach to some other tissue around the neighborhood, but most often they're attached to the thyroid gland. And that's where they get their name, because they're adjacent to the thyroid gland. Now, we want to think about staining quality and what that means for what these glands do. So just like the last one, we were talking about dark staining. Glandular tissue makes protein-based hormones, and the light staining nervous tissue is not making hormones, it's releasing hormones that are made somewhere else. Here, we're looking at tissue that has different staining qualities, and that influences what kind of hormones they're releasing. Oh, sorry, I was saying this is all the neck. So this other tissue here, this is skeletal muscle, just some neck muscle around here. And just as you're cutting through this tissue, you necessarily have to get a chunk of that there. But we're just paying attention to these 2 things here. So let's move to the higher power. Oh, actually, sorry, before we go to the higher power. Just looking at the thyroid gland here, hopefully it reminds you of a different type of tissue. If you look at this, you hopefully really are saying, oh, that looks a lot like adipose tissue or fat tissue. And there's a reason for that. If we look at it closer up, we can see, again, detail of the nuclei now, but the thyroid tissue still looks a lot lighter than the parathyroid. And that's because of these big clumps of basically non-staining material. This is pools of lipids. So if we're talking about adipose tissue, adipocytes have big lipid droplets in them. The thyroid has big lipid follicles in it. So this is a big balloon filled with lipids, surrounded by a layer of cells. So maybe down here you can see best. There's little nuclei. They're dotting all the way around here. Now, one thing that you might run across online that's a bit misleading, some sites will try to tell you that this is a simple cuboidal epithelium. That is not correct. This is glandular tissue. Glandular tissue is not a layered epithelium. But what we're looking at is a single layer of kind of square-shaped cells surrounding this lipid follicle, or making a wall of lipid follicle. The difference between that and the layered epithelium is the layered epithelium is the wall of a tube. So if we saw something like that, it would be a cross-section of tube that goes on for a long distance. Here we're talking about, we're cutting through a spherical structure, roughly spherical structure. And so at its widest circumference, it does look like the wall of a tube, but it's not a tube. It's surrounding this whole ball of lipids. And those cells, those follicular cells of the thyroid follicles, are involved in making the lipid-based thyroid hormones. So there are 3 thyroid hormones. 2 of them are just referred to as T3 and T4. T3 is triiodothyronine. I might have that name a little wrong, but the important part is the prefix is tri, as in 3, and then iodo is referring to iodine. So T3, just easier to refer to it by its abbreviation, has 3 iodine atoms in it. You need iodine in your diet for thyroid hormones to be made properly. if you don't get iodine in your diet, you develop a goiter, which is just an enlarged thyroid gland, which can get to be really big. And you can get rid of it simply by adding iodine to your diet. Anyways, so these cells are making T3 and T4, triiodothyronine, and I think T4 is called thyroxine. But they have iodine in them. They are lipid-based molecules, which means they're hydrophobic. So these big follicles are just pools of lipids, so that it's a chemically advantageous, if that's the right word, environment for this hormones to be produced. So the chemistry takes place in the lipid follicle where it's hydrophobic, so that they can be made. And then the follicular cells make thyroid transport molecules, that's not their official name, to carry the hormones through the blood, through the hydrophilic environment of the blood. But, so there's those. Now, I said there are 3 hormones, and I told you that 2 of them are called T3 and T4. Now, that might seem a little strange, because you might say, well, what about 1 and 2? There is not a T1 and a T2. It's just triiodo, etc. They name that T3, and then thyroxine they call it T4, just to enumerate. There's not a T1 or a T2. So those are the 2 lipid-based thyroid hormones. The 3rd thyroid hormone is called calcitonin. It is a protein, and it's just made, genes transcribed into mRNA, mRNA is translated into protein, you get your hormone. So that is not taking place in the follicles. That's instead taking place in what are called parafollicular cells of the thyroid. Now, actually, for this slide, I can't be positive if what I'm pointing at here is parafollicular cells, because I need more information to be positive. But parafollicular means that they're cells outside of the thyroid, which would be little clumps of cells just in patches here and there. The reason why this might not be what I'm talking about is I might be looking at the edge of a follicle just being shaved off. Not as likely, but just to be completely truthful here, I can't be sure that those are technically parafollicular cells, but for the information we have, we'll call those parafollicular cells. Or these right here, parafollicular. Or these here, these here. Anywhere we don't see a single layer of cells surrounding a lipid follicle, those are what we're going to consider parafollicular cells for this course. Again, might not be 100% accurate, but for the information we have, it's the best we can do. If we were to zoom in on that tissue, which I think I have a slide of, oh, come on. Thyroid gland 40X. Okay, so again, can't be positive, but these are most likely parafollicular cells. Or these are kind of my expectation, what I'm talking about. So there's a little seemingly lipid background and some cells there. That's probably just shaving off the very top of the follicle. That might be. But these are probably a cluster of parafollicular cells. So we have nice dark nuclei, and we have cytoplasm that's relatively dark, especially compared to the clear glass behind it. And these are the cells that are making calcitonin. So calcitonin, hopefully is obvious from the name, is involved in calcium homeostasis, or blood calcium levels, which is what the tonin in there is referring to, the tonicity of calcium in the blood. Calcitonin plays half of the regulatory role for blood calcium levels. When blood calcium levels are high, the thyroid, parafollicular cells of the thyroid, release calcitonin into the bloodstream, which tells bone tissue to take up the calcium and get it out of the blood and lower blood calcium levels. Now, the other side of that story is when blood calcium levels are low, then the parathyroid gland takes over. Do I have? Yeah. And these cells release the only hormone that they make, which is just called parathyroid hormone, which we will forever refer to as PTH as the official name of that. PTH is parathyroid hormone, which is the only hormone released from these glands, which tells bones to release calcium back into the blood and bring calcium levels back up in the blood. So these two play a little homeostatic mechanism of negative feedback to keep blood calcium levels in balance. PTH also tells the kidneys to retain more calcium and not get rid of it in the urine, and the digestive system to absorb more calcium from the food that we're eating. So other ways to increase blood calcium levels. Calcitonin does not have that effect on the kidneys or the digestive system, or the opposite effect. It doesn't tell the kidneys to get rid of more calcium, or the digestive system to not absorb as much. It just ignores those and tells the bones to store calcium, because we use the calcium to make bones hard. That's what gets it into our bones. Now, if you looked at one little patch of the parathyroid gland here and one little patch of the parafollicular cells of the thyroid gland here, they probably look identical. Sometime I need to make a picture of those two things side by side, and you wouldn't be able to tell the difference between the two tissues. That's because they're essentially the same thing. They're just your basic endocrine secretory tissue. There's no lipids. They're just making protein-based hormones. They do the exact same thing, different hormones, but basically the same thing. So when you're getting up close, the slides tend to look very similar. I guess I could have just done that. But when we pull back, the thyroid gland, because of all the lipids in the follicles, looks lighter than the parathyroid gland. And again, staining quality tells us something about that the thyroid makes lipid-soluble hormones, the parathyroid doesn't. The thyroid does also make a counterpart to PTH, which is another protein hormone, and that would be in the little clusters of cells that aren't terribly easy to make out here, but between the follicles, the parafollicular cells of the thyroid gland. So again, staining quality tells us something about what's going on here. All right, now I want to look at the pancreas. So this is not the most beautiful section. It fades off to the top and it fades off to the bottom, and that's because it's an old slide and it's just been damaged by light over the years of students looking at these in the microscope. But most of what we see in the pancreas is dark staining tissue. And actually where it fades off from the top here, that's more of this same kind of dark staining tissue. But then there are these small islands of tissue which stain lighter, and they're called pancreatic islets. So if we move to the 10x objective one, there we go, we can see one of those islands a little bit better. Okay. There are clusters of cells. We can see the nuclei there, and we can see some little nuclei in the darker staining tissue here, which this tissue is also kind of arranged in that cobblestone look that we saw in the Adeno hypophysis, the anterior pituitary. But that's not important right now. What we're interested in is the difference between these light staining patches and these dark staining patches. Now the pancreas is really a digestive organ for the most part. It makes most of the digestive enzymes that you find in your small intestine and secretes them through the pancreatic duct into the first segment of the small intestine. But it has these little clusters of cells called pancreatic islets which make hormones that are kind of related to digestive function. But really what they are the endocrine control of blood glucose levels. Now, you've probably heard of blood glucose levels and you've probably heard of insulin. Insulin is made in the pancreatic islets. But there's another hormone that's made there too, called glucagon. So just like calcitonin and PTH, these two hormones just use negative feedback to balance blood glucose levels. Blood glucose levels are high, the islets release insulin, and that tells all of the cells in your body to take out glucose from what you just ate and use it, and that'll lower blood glucose levels. When blood glucose levels get too low because you haven't eaten in a while, glucagon will tell the liver to release stores of glucose into the bloodstream and get blood glucose levels back up. Glucagon works because glucagon works on the liver because the liver stores glucose after insulin tells it to take it up in a starch-like molecule called glycogen, which you might remember from AMP1. It's found in muscle tissue also. But the liver just stores it to release it later. Also, the liver can reform glucose. So when we break glucose down, it's a 6-carbon ring, and we break it into 2 3-carbon pieces, which essentially are pyruvic acid. If we don't use those to make ATP through the chemistry of the mitochondria, pyruvate becomes lactic acid. So the liver can take lactic acid, take 2 of those and stick them back together and rebuild glucose. Now, it takes as much energy to rebuild glucose as you get out of breaking it down. So it's not really an efficient use of energy to get blood glucose levels up. But if it's the only choice you have, the liver can make new glucose, and then other tissues can use it. But hopefully glucagon is going to stimulate other aspects of getting glucose back into the system. So again, staining quality is just about the lighter staining islets, which are releasing endocrine hormones, and the darker staining tissue that's releasing digestive enzymes. Now, this is all glandular tissue. This is all making protein-based things, whether it's enzymes or hormones. The difference is just that the darker staining tissue, which we call exocrine tissue because it's secreting outside of the body, which the digestive tract is effectively outside the body, that is making a lot of enzymes to break down all of the nutrient molecules in your food. The islets aren't making that much protein because you don't need that much insulin or glucagon to make the signals that we have. Now this should be the higher power. Yeah, so this is that same islet, darker tissue surrounding it. If we were to look at this next to the parathyroid gland or the parafollicular cells of the thyroid, it wouldn't really look that different. They're basically the same kind of tissue in the end. They don't do quite the same things. They don't look identical. But this looks very light because of all the very dark tissue around it. And at the edges of these clusters of exocrine tissue, it gets really dark because this is where those enzymes are gathering to be secreted. And the protein machinery, protein synthesis machinery, is there in higher numbers. Now, there are a few different types of cells in the pancreatic islet. Alpha cells and beta cells are the main ones. Alpha cells make glucagon, beta cells make insulin. Now, we can't actually tell what are alpha cells and what are beta cells in this tissue because we haven't stained for the markers for that. They're just cells. But alpha cells are almost always in the outside edges of the pancreatic islet, and beta cells are almost always in the center of the islet. So if I were to point at this cell right here, we can't actually tell. But from what I've said about their location, this is most likely a beta cell. Or if I point at this cell at the edge here, again, can't be 100% sure, but based on what I've said about location, this is most likely an alpha cell. Alpha cells release glucagon, beta cells, like this one in the middle, release insulin. I think there's a gamma cell in there too, which is a little bit beyond my understanding of the insulin story. So those aren't the only cells, but the only ones that we're talking about are alpha cells and beta cells. And that's what we see there. So finally, the last part I want to talk about is, where'd it go? The adrenal gland. So this is clear glass here, so I could have taken a fuller frame picture of this, I suppose. But this is the adrenal gland. And the reason why I've taken it this way, actually, is this is basically the center of the gland. And we see layers of tissue as we progress out to the outside edge. Now, the adrenal gland has 2 basic parts. There's the adrenal medulla, which was covered in Chapter 15, and the adrenal cortex, which is part of the subject of Chapter 17. The reason why the medulla is covered in Chapter 15 is while it is a gland, it functions as part of the autonomic system. So preganglionic autonomic fibers, preganglionic sympathetic fibers connect to these cells and stimulate them to release epinephrine and a little bit of norepinephrine as hormones into the bloodstream. And those hormones going through the bloodstream effectively are working like postganglionic sympathetic signals to stimulate autonomic changes. We can see a very distinct line here separating the adrenal medulla from the adrenal cortex. But the cortex has a few different layers. Now the outermost layer is very obvious. There's a nice line there. But then the middle two layers kind of blend into each other. So there's lighter stuff here and darker stuff here, and an indistinct line just separating them. Now, there are three parts of the sorry, I didn't have this up ready to go. There are three parts of the adrenal cortex, and we can see them based on staining quality because they release different different hormones. So here's another picture. There is the interesting. This is labeled a little bit differently. I don't usually pull this picture up, but that's beside the point. So here's the medulla in the middle. Then there's the zona reticularis, zona fasciculata, and the zona glomerulosa. So I just need to look at this to remind myself of the names. So the zona glomerulosa is the, where'd my picture go? There we go. Is the outer part. I've always thought of it as, yeah, I guess it is. So that fibrous capsule is probably just this outer part right here. Sorry. This little outer edge here. And then this is the glomerulosa. This is stained differently than the Wikipedia page, so it's a little tough to put them side by side. But, so this should be the zona, what did I say? This is why I always have to pull it up because I get the names messed up. Glomerulosa. The zona glomerulosa makes what are called mineralic corticoids, which is a class of steroid hormone that has to do with regulating minerals. So a good example of that is aldosterone, which influences the kidney's handling of sodium as a mineral. Okay. Then the next part here, the lighter area, is the zona fasciculata, which is responsible for making glucocorticoids, which again are steroid hormones, but these are ones that influence the glucose metabolism pathways. I was hoping there was an example name here, but so glucocorticoids. And then the innermost part is the zona reticularis, back to that picture, which is this part here, and it's responsible for what I'm going to call the gonatocorticoids. So let me write this down. Okay. Zona glomerulosa, corticoid. Zona, I think that's it. What did I say? Glucocorticoids. And then the zona reticularis, what I'm calling gonatocorticoids. So, all of these hormone type names and corticoid, which is basically a word made up of cortex and steroid. So these are steroid hormones that are made by the adrenal cortex. And then the rest of the word tells you what they do. So mineralocorticoids influence minerals like aldosterone, influencing sodium levels through the kidneys. Glucocorticoids influence glucose metabolism. There's got to be an example right here, right? Cortisol, that's one. There we go. Cortisol, which is stress hormone, and therefore when you're under stress, it's kind of like fight or flight, and so it liberates energy-rich glucose for use. Okay. And then gonatocorticoids would be steroid hormones that influence the gonads, what are more commonly referred to as sex hormones. So I included this gonatocorticoid word just so that it mirrors the other ones, but most sources will just refer to them as sex hormones. And if we go down here, okay, so they don't even do that. It just produces androgens. But androgen is a type of sex hormone. It includes testosterone in its family. And it's released from the zona reticularis. Now, one last thing about staining quality to mention here. There's a vague boundary between the reticularis and the, reticularis and fasciculata. I can never do these off the top of my head very well. And when there is a structural vague thing like that, that's because there's actually a functional vague aspect of it too, which is all of this area produces glucocorticoids because the sex hormones are growth hormones, and for tissue to grow it needs energy. So this darker staining part here releases the sex hormones, but then also releases hormones to help liberate energy-rich molecules so that the growth hormones can cause growth. So there's a fading from this lighter to darker area, which is just only glucocorticoids to glucocorticoids and gonatocorticoids, or sex hormones, which is all separate from the mineralocorticoids part up here in the zona glomerulosa, whatever the name is. Anyways, and here where I've written them, if I've gotten these words at all wrong, I apologize, but the root of the word is there, the endings might be off. And I always get those messed up. But that's what's in the adrenal gland. And since I'm here, let me just say, lipid rich follicles makes T3 and T4, and then parafollicular cells of thyroid makes calcitonin, and then the parathyroid gland makes PTH, which is parathyroid hormone. So PTH is the official name we'll always give it. But now I want to point out here, parafollicular and parathyroid are often stumbling blocks for students. They see para, they figure it's this one. But the cells between the follicles are called parafollicular cells of the thyroid. Okay. And then the pancreas, there are endocrine islets, insulin, and then exocrine tissue for digestive enzymes. Oh, and I might have failed to mention this. The endocrine islets represent about 10% of the pancreas, which means that the exocrine represents about 90%. And that fits with the function. The pancreas is about 90% a digestive gland, and only about 10% endocrine. So those are the different types of tissues, and an explanation of how staining quality tells you a lot about the function of the tissue underneath. Okay, the darker staining adeno hypothesis makes its own hormones. The lighter staining neuro hypothesis is releasing hormones made somewhere else. The lighter staining thyroid tissue releases or produces the lipid soluble T3 and T4. And then there's also parafollicular cells of the thyroid that make calcitonin scattered in between the follicles. The parathyroid gland makes PTH, and these two things, parafollicular cells of the thyroid and parathyroid gland, together are the two sides of calcium homeostasis. Pancreas, lighter staining islets, insulin, glucagon, endocrine function. Darker staining exocrine tissue for digestive enzymes. And then in the adrenal gland, the medulla is making epinephrine, norepinephrine, or sympathetic nervous system. And then in the cortex, oh, shoot. What did I do? Oh, I hit the wrong button and erased it all, didn't I? Oh, that would have been so nice. Sorry. So, trying to summarize things and put... Oh, I see. Nope, that's not what I did. Nope, I just completely erased it all. I'm sorry. So, freeze frame, go back. All I was trying to do was indent the 3 layers of the cortex, so it looked a little prettier, and I just messed up by hitting the wrong button. Anyways, so let's hide my shame and go back to this. Staining quality tells you a lot about the functional aspects of the tissue, just in what's going on in the cells, what they're secreting. They appear different in the way that they stain. So, all right, thank you. That is the Endocrine Lab material.